In this video, we will discuss about another potential biomarker, that is sTRA glycans are seems to be independently expressed from the previously identified biomarkers, that is CA19. So this paper discusses about this potential biomarkers, that is sTRA glycans. So what they have discussed in this first figure is that whether they can detect these TRA glycans in the pancreatic cancer tissues, and simultaneously they also developed a approach where they want to check whether this sTRA glycans expressed at different positions than the CA19-9 glycans expressed on the pancreatic cancer tissues. So, and the antibodies that they have to detect the TRA glycans should not have these sialic acids. That is, the antibodies will not be able to detect TRA glycans if they are sialylated. And these TRA glycans, unlikely from the CA19-9 glycans, are neither fucosylated or not sialylated. So the approach that they developed to detect these TRA glycans is that first they use their approach they used to detect these sTRA glycans, that is TRA glycans with the sialic acid, is that first they use the TRA antibodies so that it can bind with non-sialic acid TRAs. Thereafter they use sialidase to trim off the sialic acids, and then they use again they use these TRA antibodies to bind to this sialidase treated TRA. In the first treatment they use the labeled TRA so that they can distinguish the newly bind TRA and the previously labeled TRA, and thereby they will be able to detect the sialylated TRA glycans in the samples. So this is the approach that they used. And in the panel B they shows the similarities between CA19-9 and sTRA, where we can see that in the case of this CA19-9 they have this type 1 leg neck, whereas in the case of sTRA they have both the type 1 leg neck and the type 2 leg neck. And first they check this pancreatic cancer tissues for the presence of this TRA166. So when they do not use any sialidase, they do not see too much signals for these TRA glycans. However, once they use the sialidase treatment and after that they use the TRA160, they can see that the presence of this TRA glycans, indicating that in the pancreatic cancer cells most of these TRA glycans are sialylated. Thereafter they developed the approach to distinguish between or to check whether these CA19-9 glycans or the sTRA glycans are expressed at the same cell surface or at the same positions or not. So approach is mentioned in this panel D, where they used this hoist dye to stain the nucleus of the cells, and thereafter they use this anti-protein 1 to stain the proteins. There is a green fluorescence to stain the proteins, and TRA monoclonal antibodies to stain the glycans, or antibodies to stain the glycans. And thereafter they did the scan fluorescence and then quench the fluorescence and use the same tissue sections for the another approach, where they use the same hoist dye and this anti-protein 2 against the second protein of interest, and thereafter they checked it for the CA19-9 monoclonal antibodies. After that they scan the fluorescence and quench the fluorescence, and thereafter they treated it with the sialidase and then use the same antibodies that they used in the first step, that is host antibodies and the anti-protein 3 and this TRA monoclonal antibodies. So the anti-protein 3 looks like similar to the anti-protein 1, and we will discuss that thing in subsequent videos, that what does the anti-protein 1, anti-protein 2, anti-protein 3 means. So for now they use the anti-protein 3 host and again these TRA monoclonal antibodies to check what are the sialidase treated glycans are there. So in this case they will be able to see the sialidase treated glycans, and thereafter they can compare these three results to see whether the CA19-9 overlaps with TRA monoclonal antibodies, that is without sialic acid, or TRA monoclonal antibodies with the sialic acids. So this is the approach that they have used, and in this panel 3 they will follow this step where they use this TRA labeled monoclonal antibodies before sialidase treatment, and then TRA monoclonal antibodies after sialidase treatment, so that they will be able to see the sialylated TRA monoclonal antibodies distinctly, and can be able to differentiate them from non-sialylated TRA glycans. So I hope you understood their approach and some of the results that shown in the panel C. And if you need to understand more about their sTRA glycans and how they come up to this idea, whether this could be potential biomarkers for pancreatic cancer cells apart from the CA19-9, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.